Hello, everybody. This is Dr. Karen's again, and I will be discussing just a small part of Chapter 11 for microbiology for non-science majors at Eastfield. So, like I just said, we're just going to cover a small amount of things in this chapter. Some of which is going to talk about the shape or morphology of bacterial cells. So morphology means shape, and here we're talking about the shape of the actual cells. Later on in the semester, we will talk about the shape or the morphology of bacterial colonies, which are big collections of cells, masses of cells, but that is something different. So we're just looking at the individual bacterial cells here. And I mentioned in chapter 3 that it is the cell wall of bacteria that contributes to that morphology or that shape. And that is true. And depending on the species of the bacteria, that will dictate the morphology of that particular cell. So here are examples of different prokaryotic or bacterial cell morphologies. The two that we're going to see in the lab are coccus, or cocci is plural, and that means round or spherical. And then bacillus, or bacilli is plural, and that means rod-shaped. So most of the bacteria that we're going to be working with are going to fall under one of those categories. And with just a simple staining technique, you can see those different shapes really easily under the microscope. Some of the other morphologies that unfortunately we won't get to see: coccobacillus, that's like a short, fat rod, not quite round, not quite as long as an actual rod. Vibrio, which is more like a comma shape or almost sometimes like a V shape a little bit. Spirillum and spirochete, which are both a little bit corkscrew. Spirochete more corkscrew than spirillum. When you see the word pleomorphic, and you see like what kind of shape is that? I have no idea. There are some species of bacteria that don't have a consistently defined shape. The cells can take on any shape, and that's what we mean by pleomorphic. And then there are some prokaryotes that have this star shape. But we're going to focus again mostly on coccus and bacillus. Chapter 11 brings up endospores again. So in chapter 3, we kind of looked a little bit about how they formed. They talk a little bit more about them here. To reiterate, Bacillus and Clostridium, these are two different genera of bacteria. They're gram-positive. They are the ones that can form endospores. Nothing else can. So they show you a picture here. If you were to stain, these are two different types of stains. This is the type, if you take the face-to-face class with me, we will actually do this endospore stain in the lab. But if you're on the online class, we won't. The green are the endospores. So sometimes you can see the endospores outside the cell. Sometimes you can see them while they're still inside the cell. And here, in the image on the left, I think in this particular staining technique, all of the endospores are still inside the cell. All right, so that was endospores. Chapter 11 talks briefly about how prokaryotic cells reproduce. So in terms of bacteria, and some of the other prokaryotes, and even some eukaryotic microbes will do some of these types of cell divisions as well. But for bacteria, it's mostly going to be binary fission, which is basically mitosis. I mean, it doesn't go through the phases of mitosis like you would see in a eukaryotic cell, but the end result is the same. You start with one cell, you end up with two identical daughter cells. But in this case, you know, you're not going to see metaphase and anaphase and all that because the chromosome is not nearly as organized as they are in eukaryotic cells. But basically, one cell splits in half. Snapping division is the same type of thing, but the two daughter cells fail to completely separate at the cell wall, and that they kind of end up kind of straggling onto each other, and you get like this hinged look of these two cells, and that's where this term snapping division comes from. Not all cells will do that when they divide. And then budding. So this is going to be mostly for yeast. We're going to see budding. So instead of where we saw in binary fission, there was one cell that split in half to form two new cells. In that case, there was no parent cell left at the end. The parent cell was gone, and you ended up with two daughter cells. In budding, that is not the case. In this case, the parent cell remains. The DNA that is duplicated kind of buds off into a new daughter cell that's small to begin with, and then it will grow and get bigger. But the parent cell is still there and can probably bud again and create another daughter cell at another time. So we'll see that mostly with yeast. All right, so going back to bacterial cell division, that snapping division and that binary fission, depending on what plane the division occurs on through the cell and how many planes division will occur at the same time, that will lead to what we call different arrangements of the cells. So let's show the pictures to give you an example. So here we have one coccus-shaped cell, one round cell. When it divides along one plane into two cells. If those two cells stay very close to each other, when you see them under the microscope, you're seeing groups of two cells. That is the arrangement of the cell, and that arrangement of two cells is diplo. Because the cells are coccus in shape, we call that diplococcy. If that division continues, you're going to end up with a chain of cells. The chain is an arrangement, and that is referred to as strepto. We call this here streptococcy because we have a chain of round cells. Tetrads is when there's division along two planes. You get 4. You keep adding planes. Now we have like cubes of cells. That's called sarcinae. And then if you've got multiple planes of division happening on one cell in all different directions, you're going to end up with what's called kind of this bunch of grapes arrangement. And we call that staphylo. So staphylo is bunch. Coccy because the cells are round. Bacilli cells have similar arrangements. You can have a single bacillus. You can have two bacilli cells together. That's diplobacilli. Streptobacilli would be a chain of rod-shaped bacteria. And there's no staphylobacilli. Instead, there might be palisade, which is like a zigzag chain, or V-shape when you have, this would be the result of snapping division. Two cells that are still kind of hinged together. So you should be familiar with not only what the shapes look like, what bacillus looks like, and what coccy looks like, or coccus looks like, but what these different arrangements are. If you were to see a picture, could you figure out what the arrangement is? Do you know that this is streptobacilli? If I were to give you a description of the arrangement and the shape of the cells, can you tell me what that term is? So practice with these images. You can look online for lots of other images too, and just kind of get used to distinguishing among them when you're looking at microscope images. That honestly is pretty much it for chapter 11. The rest of this chapter talks about all the different groups of archaea, all the different groups of bacteria, and things like that. If you look at the study guide for the first lecture exam, part of what you need to know for chapter 11 is I give you a list of bacteria, and I'm asking you to tell me which ones are gram-positive, which ones are gram-negative, and what the morphologies are of those bacteria. Are they coccy? Are they bacillus? What are they? So you can find that information in chapter 11, and then I also will post an answer key so you know you have the right thing. So use the rest of this chapter with the study guide to kind of figure out what those different bacteria are in terms of gram stain and morphology. Other than that, that's it for chapter 11.